purification. DNA can be prepared from a whole range of materials, including blood, exfoliated bladder cells, frozen tissue samples, and paraffin tissue blocks. Since DNA is generally stable under suitable storage conditions, it can be prepared and stored in batches. DNA extraction and purification involves three basic steps: breaking the cells open to expose the DNA, removing the membrane lipid through the use of an appropriate detergent, and precipitating the DNA with alcohol. There are also two optional steps that are almost always done prior to the precipitation step: the removal of protease by adding an appropriate protease, and the removal of RNA with the use of an RNase. Through DNA purification, our samples, you can minimize the disturbance from non-target DNA and other contaminants and increase the stability of your sample in long-term storage. RNA extraction and purification, on the other hand, involves four basic steps. First, you need to distribute the cells by adding 1-didyl thiosinate and the reducing agent to the sample and the subject into vigorous shaking or vortexing. This step will also break the disc-full head bonds and inactivate the contaminant proteins present in the sample. After cells lysis, you need to add phenol and chloroform isoamyl alcohol to separate your RNA sample from the solution. The echo phase contains your precious RNA, so you need to put it in the separate tube. Add isopropanol and centrifuge the solution to precipitate your RNA. Washing the precipitate with 75% ethanol will remove any impurities from your sample. However, it is important to note that RNA is less stable as compared to DNA, so you should only isolate it prior to use. Special care should also be taken before, during, and after the process to ensure the purity of your sample. Remember, even the smallest amount of RNase can compromise its quality. DNA and RNA can also be isolated from the same biological sample by extracting a total nucleic acid fraction and dividing into two parts, one of which will be treated with DNase, while the other portion will be treated with RNase, to recover RNA and DNA respectively.